Hello, my name is Mario Hernandez, and my target audience for this recording will be high school students. So I will be starting this presentation off with how to define a virus. So, what are viruses? Viruses are these nanoscopic particles that surround us. They are in the air, they are in the dirt, they are in our skin, and they've been around way longer than humans or even dinosaurs. And that can even be traced back to the Paleozoic era, which is hundreds of millions of years ago. So we see here you have a nanoscopic virus, and here is a host cell. Viruses are obligate parasites, meaning they need a host in order to reproduce. Viruses are generally made up of two components. There is the capsid and the viral genome, which is nucleic acid. The capsid is generally there to protect and transport the viral genome. It surrounds the virus or the genome and is composed of a number of protein subunits known as capsomeres. While this genome contains the blueprints of assembling more of itself, kind of like human DNA, as you would say. So, viruses are classified in a few ways, and one of these ways is its size. Compared to, or the introduction to electron microscopy has given virologists a way to visualize these viruses and are able to see them with the naked eye. So, on the left here, we have a rotavirus, which symptoms may include watery diarrhea, vomiting, fever, and other abdominal pain. So you can see that this rotavirus was acquired through a fecal sample from a child. And so on the right, we have bacteriophages that are surrounding this host bacteria. And you can see how small they are compared to the bacteria. There are about 20 to 30 individual particles on just this one side. Another way that we classify viruses is by its shape. So after the introduction to the electron microscopy, we can see how viruses are shaped. So I would say the first one that is shown right here is helical shape. An example of this would be the tobacco mosaic virus, which is a plant virus. It wraps around in a long strand-like structure. The second is a polyhedral or isohedral shape. And you can see here that they have 20 triangular faces. They're all equilateral to each other. An example of that would be adenovirus, right? A third one shown here is a more spherical shape like influenza. And the last would be a more complex shape that is a combination of both helical and isohedral. So here we have a bacteriophage right here, and it has a isohedral head with a helical tail, as you can see. The third way that we classify viruses is of an absence or an inclusion of an envelope. So if you notice that there are two viruses here, the right one has just the capsid and the genome. In other words, an unenveloped virus, while the one on the left is an enveloped virus. As you can see here, they are enclosed with a lipid bilayer, which in fact they steal from their host cells and help them infect and enter other host cells. They cannot produce this bilayer by themselves. Last but not least, and the more recent one, would be classifying viruses by its genome. In total, there are 7 different kind of genotypes in all viruses. And all 7 of these have just one goal, and it is to convert the viral genome into a template that the host cell can read and follow its instructions. This template is mRNA. This center one here is the goal, is to is the template, and it allows the host cell to start assembling all the parts required to build more virus particles. The goal is just to replicate and make more copies of itself, and depending on the kind of genome, the virus has different strategies to reach that end goal. We got double-stranded DNA, which is something we humans also have. There are some that are just a single strand of DNA going from left to right, being positive sense. The other one is we'd call it negative sense DNA, which is just a single strand, but it's the complementary strand of the positive, so it's going from right to left. And we also got viruses that are RNA based, either double like the DNA or single stranded positive or negative sense. So that is the most recent way or newer way that scientists are starting to classify these viruses. So in the end, why do we study these viruses? Well, because they're everywhere. If we were to stack every virus from one end to another, it would reach 200 million light years, which is basically double the size of the Milky Way galaxy. And that's a lot of them. Normally, these viruses are generally looked at as these extremely dangerous particles that cause only disease. It may be the case for some, but not all are infectious or cause disease. Some are beneficial to humans. For example, mice can acquire a resistance to Yersinia pestis, which is plague, after being exposed to a form of murine herpes virus. And what that means is that those specific mice cannot harbor or have any plague virus, and they are therefore not a vector. I came across this journal, Viruses, which was talking about how scientists were using these bacteriophages to, in turn, make these bacteria that are already becoming more and more resistant to antibiotics, more susceptible, or going back to being susceptible to those same antibiotics. So here we have Pseudomonas aeruginosa bacterium, which is more commonly found in natural sources of water like lakes and rivers, and is one of the more common bacteria that is tested for in drinking water. So by selecting certain bacteriophages that bind to this multi-drug pump, it is possible to restore the sensitivity to several antibiotic drugs. These phages can also be paired with antibiotics that inhibit certain select bacterial processes that are not needed for the bacteriophage production. Kind of like a synergy interaction. Another journal that I read, I saw that scientists used the cryo-electron microscopy to structure out the Zika virus, and you can see here all the individual glycoproteins in red, and also the cross section. So using these kinds of techniques, other scientists are able to map out other viruses. For example, using this same technique, scientists can use it for looking at the mode of action for HIV that we have down here, that we can see all the receptor or the glycoproteins. And it's not just HIV that we can use this for. It can be used for influenza or currently SARS-CoV-2. So with that kind of technology, they're able to map out and illustrate how these viruses can enter our host cells, immune cells, or specific cells that these viruses target, eventually leading to these antivirals that target those receptors and stopping the virus from infecting cells. So thank you for listening to my presentation. These are my sources cited that I use in this presentation, and thank you.